Hello, good afternoon. I think we can start already. So it is my pleasure to introduce my colleague, Dr. Gael Roué. He received his PhD degree in molecular and cell biology in 2002 at the University of Caen in France and worked as a postdoctoral fellow at the Institut Pasteur on B-cell malignancies. Then he moved to Hospital Clinic of Barcelona, Spain, where he participated in the characterization of new therapeutic agents for chronic lymphocytic leukemia and mantle cell lymphoma. Between 2008 and 2017, he held a tenure track position at the Hematology Oncology Department of the IDIBAG Research Institute in Barcelona, focusing on B-cell lymphoma. Between 2017 and 2019, he co-supervised the molecular genetic unit of the hematology department at the Vall-d'Hebron Hospital here in Barcelona also, and continued his research in B-cell lymphoma, hosted by the experimental hematology group at the BIO. Since November 2019, he leads the Lymphoma Translational Group at the Josep Carreras Leukemia Research Institute here in Barcelona, with a special interest in applying multi-omics approaches to unravel the interplay between oncogenic signaling, B-cell receptor pathway, and intrinsic protein homeostasis in the context of lymphoma stroma crosstalk. So before the talk starts, just remember to leave your question in the chat so that I can handle them to the speaker at the end of the session. So Gael, nice having you here. It's a pleasure to be here in your seminar, and you can start, I think, whenever you can. Thanks.
Okay, so thank you, Ruth, for this kind introduction. So it's a pleasure for me to participate in this new webinar series organized by AGC. Today, I will present unpublished data we have generated in the lab in the last 2 years, focused on a new line of investigation for us, which is the targeting of immune checkpoint in aggressive B-cell neoplasm. So this is a brief outline for this presentation. First, I will remind you some main facts about the immune landscape and the response to immune checkpoint blockers in aggressive B-cell lymphoma. We'll mostly focus on the CD47 therapy, which is a very promising therapeutic target in B-cell neoplasm, including aggressive B-cell lymphoma. We'll also discuss about the possibility to increase the response to CD20 antibodies with the anti-CD47 treatment by a process called optimization. And then I will present briefly some field projections for this project in the lab based on the development of new preclinical model able to modelize the 3D structure of the lymphoid tumors. So this is a picture you know pretty well, showing the main B-cell lymphoma, the main B-cell malignancies we are working on in the lab. Mostly, yeah, two type of disease that derive from the germinal center or from the mantle cell of the lymph node. And in the last 2-3 years in this model, we have been mainly focused on using epigenetic drugs targeting BRD4 to promote the protein, since this kind of therapies are able to counteract the crucial signaling within this B-cell malignancies, mainly focusing on mixed signaling, BCL2 and NF-κB genes. And this kind of inhibitors, combined with different other biological agents interacting with BCL2 proteins like venetoclax, the chemokine receptor 604, high Iraq in the mid-88 mutated cases of ABC, DLBCL, but also in combination with the current treatment for multiple myeloma, Lenalixa. So we have seen a very nice interaction of this kind of approach with these biological agents. But it came out some years ago that when we were focusing on MYC activity in all of this model, but MYC was also involved in the creation of another kind of immune regulators, which are the immune checkpoints. So in this paper published in Science in 2016, the authors described in a Xenoblast Model of T-cell acute lymphoblastic leukemia but the depletion of MYC could induce a very nice decrease and blockade of tumor border in these mice. But when these tumors overexpressed CD47 and PD-L1, we can almost counteract this effect at both tumor size and minimal residual disease level. So this negative activity towards the MYC downregulation were related to the regulation of tumor-infiltrating lymphocytes, the T cells, which entry into the tumor were blocked by CD47 and PD-L1 in this tumor. So, more generally, it is known that in most of the tumors we have the activation of MYC transcriptional programs implied. So, involved in the relation of PD-L1 and CD47 blocking signaling, making that these immune cold tumors, the so-called immune cold tumors, are characterized by low infiltration of T cells and increased infiltration of tumor-associated macrophages, TAM cells, and the myeloid suppressors. So we know that by targeting different epigenetic regulators that can affect the MYC signaling, so promodomain inhibitors may be such agents, we can obtain the immune host tumor, hot tumor, where the infiltration of T cells is increased and the interaction between the tumors and TAMs is blocked by the restoration of the blockade of a PD-1 therapy, the increased response related to interferon, and this could allow to the combination of epigenetic drugs with different kind of immunotherapeutic blockade, the immune checkpoint inhibitors. When we focus more on B-cell lymphoma, so the cold tumor and hot tumor could be named non-inflamed lymphomas and inflamed lymphomas. So in the non-inflamed lymphoma, which correspond mostly in those lymphomas with activating mutation of P10, EZH2, P53, or MYC, so the tumor is largely infiltrated by TAM cells, whether in the inflamed lymphoma we have the presence of TAM, but also CD4 positive, CD8 positive T cells, which activity is more blocked due to the derivation in the expression of the PD-1 ligands, PD-L1 and PD-L2, but also in the defective interferon-dependent signaling, putting out the possibility to target PD-L1 and CD47 in these tumors by specific blocking agents. In fact, this therapy have been developed in the last decade. So most of the agents that have been entered in the clinic so far are targeting a PD-1 or PD-L1 on the surface of the tumor, but also CTL4 or CD18. So these are the main kind of drugs or small molecule inhibitors that have been entered in the clinical in refractory lymphoma patients. But we have also some small molecule inhibitors or blocking antibody targeting CD40, CD47, CD80, which are being analyzed in these settings. So you can see here the increase in the interest, in the clinical interest in targeting such a molecule in the last 3 to 4 years. So a couple of years ago, there was a huge increase in the number of PD-1 clinical trials in advanced stages in phase 2 of phase 3 clinical trials. And in the alternative targets that are being analyzed in the last 2 or 3 years, most of them are focused on CD47. So why for CD47? So you know that this immune checkpoint corresponds to this don't hit me signal, which is expressed on the membrane of the tumor cell. So these receptors block the signaling by CYP-alpha at the surface of the macrophages. So it blocks the recognition of macrophages by of the tumor cell by macrophages, blocking the phagocytosis of these cells, and the targeting of CD47 by blocking antibody here in yellow could block these signals and allowing macrophages to recognize tumor cells and leading to the engulfment of the cells by macrophages. Another effect of such antibodies is to promoting the FC receptor response in natural killer cells, leading to increased sensitivity or increased possibility to liberate the perforin enzyme that could lead to the death of the tumor cells. So what about in aggressive B-cell lymphomas? So if we take the example of mantle cell lymphoma, the tumor microenvironment is pretty well known. So we know that the tumor cells receive support from different kind of cells, from accompanying T cells, follicular dendritic cells, mesenchymal stem cells, and they receive input from very various signals that could help in the proliferation, in the resistance to drug treatment, and the migration of the cells. So here you have the different signaling pathway, which are mainly based on B-cell receptor pathway, NF-κB activation, AKT signaling, downstream of different membrane receptors, and following the stimulation of this pathway by accompanying cells, which can be either T cells or mesenchymal stem cells, follicular dendritic cells, or endothelial cells. But previously, while we know we know most of this signaling pathway involved in MCL resistance, drug resistance proliferation, we don't really know what is the CD47 status in this model. Conversely, in other aggressive B-cell lymphoma and DLBCL, we know that the expression of CD47 is associated with worse prognosis for the patients, but we don't really know which are the main component of the tumor microenvironment that could be involved in the progression of the disease, in the resistance to treatment, and so on. So to treat this aggressive lymphoma, some antibody have been introduced in the clinical practice many years ago. So you may remember the antibody produced by Stanford University and B6H12, presented some weeks ago by Dr. Weissman. With this antibody has shown for years activity in preclinical model of acute leukemia, of lymphoma, and has not entered in the clinical, but it rapidly showed some side effects related to the hemagglutination of the red blood cells. Since the CD47 is a ubiquitous receptor which is highly expressed by platelets and red blood cells. So to try to improve on the response and to lower the adverse events linked to CD47 therapy, a new set of antibody have entered the clinical a few years ago. For example, this one from Celgene and this one from Trium. And results will be published in the upcoming years. In the lab, we started to work with another antibody, which I cannot disclose the origin, but it's also from M Oncology company. So the antibody we used in preclinical model of lymphoma shows some activity as a single agent. So you can see here the activity in terms of killing of the tumors. So we are using here cell lines from a Burkitt lymphoma, Raji. So there is some activity either alone or in combination with rituximab, which is an anti-CD20 antibody. But most of this synergism between the two therapy was observed in diffuse B-cell lymphoma. So in these cell lines, we have a very nice increase in the killing of the tumors by combining both CD47 blockade and CD20 inhibition. This was also the case in primary samples. So here you have a very nice test rate observed in this patient sample. Surprisingly, when we extend this analysis to a set of different primary culture of TCL, DLBCL patients, we don't see an increase in the phagocytosis of the cell. So this is the basal phagocytosis observed with rituximab, and we combined the two antibody. The effect was almost the same. There was no difference between rituximab or rituximab plus CD47 treatment. And this was mainly due by the fact that by direct immunofluorescence, we observed that calcium-loaded DLBCL cells treated with the two antibody do not really enter within the macrophages. So you have here macrophages studied in the 96 plates, and all the tumor cells remain like at the surface of the macrophages, they fail to enter within the macrophage to be processed. So in an effort to try to improve the response and the activity of this therapy, we started to work a couple of years ago with TG1801, which is a bispecific CD47 CD19 antibody developed by Novimmune and TG Therapeutics. So this is a kappa lambda bispecific antibody able to block CD47 signaling on the surface of B cells. So as you can imagine, this can avoid the adverse effects toward the platelets and red blood cells in both in vitro and in vivo models. So this antibody has also demonstrated the capacity to facilitate the engagement of effector cells like macrophages. So it can increase the phagocytosis of cancer cells, and also it could increase the antibody-dependent cell cytotoxicity related to clinical cell activity, both as single agent or combined with the CD20 therapy. So why CD19 bispecific antibody? So this is mainly because CD19 expression is almost maintained during all the differentiation program of B lymphoid lineage. So, and as you can see in the B cell lymphoma, this expression is really high and maintained within the different modes. So in preclinical setting, this antibody is showing a very nice activity in terms of phagocytosis induction. As you can see here, this antibody in total is much more active than the combination or of two monovalent CD47 or CD19 antibody, either used as a single agent or in combination. So a really nice activity by targeting CD47 on the surface of B cells. In vivo settings, in mice grafted with B-cell lymphoma, this is the effect of rituximab single agents on the control of tumor volume in these mice. When we use a CD47, CD19 antibody, there is significant activity, but almost all of this effect is synergistic when you combine the two antibodies. And as a consequence, and as expected, the survival of the mice is almost completely preserved in the case of the combination group, compared with the single agent arm here in the CD47 alone, rituximab alone, or the vehicle. So in the lab, based on this data in the lab, we were interested in analyzing the activity of TG81 in a technical model of non-Hodgkin lymphoma, mainly mantle cell lymphoma and blockade lymphoma. So this is the work of Marco Rivero in the lab. So this was part of the project. Marcelo focused on the establishment of phagocytosis and ADCC assay in the lab. For the second part of the project will be to analyze the antigen presentation and the direct tumor cell apoptosis with this antibody. So regarding the first part of the project, so we were interested in combining CD47 blocking with anti-CD20, and for this, we focused on obinutuzumab, which is a new generation glyco-engineered anti-CD20 antibody produced by TG Therapeutics, the provider of the CD47 antibody. So this antibody is showing very nice response in different B-cell malignancies and is actually in phase 3 pivotal trials. And in combination with obinutuzumab, the TG Therapeutics has introduced some years ago PI3 kinase delta inhibitor in umbralisib. So this inhibitor is showing similar activity when compared to idelalisib, which is the first-in-class PI3 kinase delta inhibitor. But most importantly, is showing fewer adverse events when compared with idelalisib. And the combination of obinutuzumab and umbralisib, which is called the U2 regimen, is showing very nice activity in different diseases. So here you have the results in refractory relapsed DLBCL patients, where we observe a complete response and not complete disappearance of the tumors following a few cycles of treatment in refractory patients using the combination of two agents. So this combination is useful to block the B-cell receptor pathway, which is a crucial pathway involved in B-cell lymphoma development. So the BCR is here is It involves the phosphorylation of a series of downstream kinases. So, almost among these kinases, the Pluton tyrosine kinase, BTK, is maybe the most important one, and has been the focus of different therapy directed at blocking its activity. So the first BTK, the first in-class BTK activity inhibitor, ibrutinib, is now approved by FDA for different B-cell malignancies, and it is the case for acalabrutinib, which is a second generation BTK inhibitor. So downstream of these kinases, there is a series of signaling cascades involving AKT, mTOR pathway, NF-kappa B, and HERG, which leads to the proliferation of B-cell differentiation and inhibition of drug sensitivity. To block this pathway, so beside BTK, some drugs have been designed. So here you have the idalicib I commented previously. So we could say that at the same level, we have umbralisib blocking PI3K signaling. And also, at least in mantle cell lymphoma, some mTOR inhibitor have been approved, like everolimus in Europe, for example. So when in the clinical, this combination of the new generation CD20 rituximab and umbralisib, this is a U2 regimen, is showing very nice activity in combination with BTK inhibitor ibrutinib in refractory CL patients. So here you have the overall response of this triplet when compared to ibrutinib single agent. So sorry, in this case it's only rituximab. So when comparing with everything, there is a very significant increase in the response rate in these patients. So the first endpoint, primary endpoint of the clinical trials is being reached so far. And in the triple combination assays, combining U2 regimen plus ibrutinib, we are seeing a very nice response in almost all of the B-cell entities considered in this trial. So you observe a 90% response in mantle cell lymphoma, including syncytic leukemia, small lymphocytic lymphoma, marginal zone lymphoma, follicular lymphoma, and in a subset of DLBCL patients. So this trial validates the use of U2 in combination with BCR inhibitor in B-cell lymphoma patients. So to try to validate this therapy, this kind of approach in vitro in a model, so before combining with the CD47 blocker, we first try to validate U2 regimen as a possible synergistic treatment with BTK inhibitor. So for this, we used TG1701, which is a new generation BTK inhibitor developed by TG Therapeutics. So it has an increased selectivity towards the BTK, as you can see in the genome 3. And in the clinical, is this BTK inhibitor is showing a very nice combination activity with U2 in different B-cell entities. So for this project, in cooperation with the group of Pablo Menendez and Meritxell Viñolas, we generated a recurrent mantle cell lymphoma cell line harboring the mutation in the ibrutinib BTK binding pocket in BTK. So this mutation, C481S, leads to a decrease in the cell response to ibrutinib, as you can see here. In the standard dose, there is almost no response to ibrutinib, while in the same mutant cells, where compared to the parental cells, the response to TG1801, this challenger BTK inhibitor, was almost maintained with no substantial increase in the GI50. When we use a second model of resistance to BTK, so this is also an MCL cell lines, which has been generated, treated for 8 months to ibrutinib, and which has developed a resistance link not to mutation in BTK, but in the activation of non-canonic NF-kappa pathways. So this model in vivo is almost resistant to ibrutinib. As you can see here, there is no significant decrease in tumor growth. And this model is also resistant to TG1701. But what we observe in this tumor is that there was an increased infiltration of CD56 positive cells, NK cells, and also an increased infiltration of F480 cells, which are macrophages. So this effect was significantly higher with TG1701 when compared with ibrutinib. So it was quite interesting, and one of the important differential effects between 1701 and ibrutinib was the combination activity with anti-CD20 antibody. So as you can see here, we are evaluating the ITCC activity of CD20 ligation in the case of rituximab, but it was also somehow the same case with rituximab. We observe that while ibrutinib inhibits part of this ITCC activity, when we use 1701 in vitro, we can maintain ITCC activity following rituximab treatment, and in a lower extent following rituximab treatment. So based on these results, we used this triple combination in the BTK-resistant mouse model, and you can see here that there was a very nice reduction in tumor growth when we used this combination when compared to the TG1701 alone. And this effect was related to an increased inflammatory pattern in this tumor. So we detect by targeted PCR an increased expression of IL-2, TNF-alpha, CCL3, and a lower expression of the anti-inflammatory cytokine interleukin-10. And in this tumor, we also detect an increased infiltration of NK cells. So once we validated this therapy, this U2 regimen in a preclinical model of non-Hodgkin lymphoma, we decided to use this combination together with CD47 blocking antibody. So for that, we use this complex cookie system, associating tumor B cells with the PBMC as a source of NK cells and T cells, stromal cells, and M1 polarized macrophages. So in these settings, we observe a nice activity of rituximab single agent. This is like a control activity described pretty well in different B-cell lymphoma. But when we use rituximab, we see a significant increase in phagocytosis mediated by macrophages, and also a significant increase in ADCC mediated by NK cells. But most interestingly, when we use rituximab in combination with CD47, I mean activating CD20, blocking CD20, and blocking CD47 signaling, there is a significantly increase in ADCP induction in this model. And it was almost the case when we used rituximab plus ibrutinib plus umbralisib combination with TG1801. We also observed a significant increase in ADCC, so arguing in favor of combining CD47 blockade with CD20, both for increasing macrophages and NK activity in B-cell lymphoma. So to try to decipher the mechanism, intrinsic mechanism related to the synergistic activity, we made a RNA-seq analysis at CENAG of the U2 receiving tumors versus the control group, and we also compare the U2, umbralisib plus rituximab versus CD47 blocking antibody plus U2. And since with U2 we don't see much pathway activated or regulated with the blockade of PI3 kinase delta and the activation of or ligation of CD20, mainly redox activity. In the case of the triple combination, we observe a downregulation of many pathways associated with cytoskeleton remodulation, with membrane or cell membrane, and with response to stress. Here you have the top 20 geontology terms detected by this analysis. And you can see that the cytoskeleton is the fourth of them. We were able to validate the polymerization of F-actin as a biomarker of activity of this combination. Since in the control tumors there is a high degree of F-actin polymerization, but when we treat with U2 plus or 18-1 or U2 plus 18-1, there is a synergistic decrease in this polymerization. So this is quite interesting because seminal work focused on CD47 signaling in B-cell malignancies mainly chronic lymphocytic leukemia. I've described several years ago that the ligation of CD47 by an immobilized antibody was able to activate some specific cytosolic proteases, the surplus family of proteases involved in the destabilization of F-actin filament within the cytosol. So this depolymerization of actin leads to a cytosolic release of DAP1. So it is important because DAP1 is associated with the regulation of mitochondrial fission and mitochondrial fusion. So this leads to the remodulation of the mitochondrial membrane, leading to mitochondrial depolarization in the absence of cytosolic release of apoptogenic factors. So you know that within mitochondria, a set of proteins are usually released to induce classical caspase dependent or caspase independent cell death. So these factors like cytochrome C, apoptosis inducing factor, SMAC Diablo, endonuclease G, or Hb. And these factors were not released within cytosol of cells treated with anti-CD47. But the main hallmark of this was the ecto exposure of phosphatidylserine. So the specific type of cell death, which is subtypes of caspase-independent cell death, was called type 3 program cell death in CLL. So if we come back with our model, we used in vivo the combination of U2 plus CD47 antibody. So as you can see here, the single agent treatments are very effective in this tumor. So we observe almost 80 to 90% of tumor growth inhibition, either by blocking CD47 or by blocking PI3 kinase delta plus CD20. So you may say, what is the advantage of treating the mice with this triple combination? But the main advantage was that 1 month after stopping the treatment, so when you the number of the relevance, the percentage of tumor-free animals, so this percentage was maintained up to 40 to 45% in the case of the triple combination. So this was the main advantage, that is that you can stop the treatment, and then most of the animals remain free of tumors several weeks after stopping the treatment. While when you stop to give the treatment to the animal, when we use another arm of treatment, most of the animals present lower tumors and some tumors that are increasing in volume after stopping the treatment. And this effect by histological examination was mostly related to an increased infiltration of macrophages, as you can see here. So as expected, the CD47 antibody induced infiltration of tumors by macrophages, by mouse macrophages. So it was also the case with U2, and you combine the two drugs, there is a synergistic tumor infiltration by mouse and macrophages. In the case of NK cells, it's not so clear, so we are not sure whether the promotion of NK infiltration is so relevant for this model. From these different tumors, we made a targeted qPCR array to detect some crucial genes related to cytoskeleton remodeling, to cell cycle and inflammation. And we observed that with the combination treatment, there is a decrease in beta-actin expression, as you can see here. There is a decrease in genes related to cell cycle regulation, like cyclin D1 and E2. But most interestingly, there is an increase. There is like a pattern of pro-inflammatory context with an increased expression of IL-2 that we observe there, and IL-6, and some lowest level of IL-10. So the perspective for to validate these findings in vivo, so in vitro, so will be used original 3D organoids model that we are developing in the lab. So this is the work of Gina Santos and Mark Mengol in the lab. So they are working through the European project Proteoblood through the development of a new organoid 3D model of B-cell lymphoma, mainly aggressive B-cell lymphoma. So in these models, we are co-cultivating either primary or cell lines of B-cell lymphoma with macrophages, macrophages, with strong cells. The cells are labeled with a nanoshuttle. We are applying a magnetic field on this culture, and this allows us to obtain like a 3D structure growing in vegetation in the culture plate, and this structure may show some aspect of cell-cell interaction, some regulation, some established red between the different cell types. And this is going to be analyzed by histological analysis and omics like proteomics and multi-genomics characterization. So here are the first preliminary data we have obtained in this model. So in this case, we are cultivating mantle cell lymphoma cell lines with embolized macrophages and with a follicular cell line, HK. And you can see that after 3 weeks, we are conserving structure associating B cells. So within the core of these organoids, we have B cell interacting with M2 macrophages here in red and B cells in green. There is a strong interaction, while it seems that stromal cells are answering, let's say, the formation of the organoid membranes here at the periphery of the organoids. So we are currently analyzing the histology of these tumors, and we will soon have the first samples for advanced proteomic and genomic analysis. So the other models we are going to work on in the next months, or only next year, is to develop an in vivo model of B-cell lymphoma. So for this, we'll use chicken embryos, Xenoblast, which has been developed for many years in a different group and as well in the private company. So this model consists in injecting the upper choreo-oriented membrane of fertilized chicken embryos on the upper side. So you can inject the tumor there at the day 9 after fertilization, and it allows you to study the development of the tumor for about 10 days, which is more than enough because thanks to the high vascularization of these models, you can reproduce in 10 days something that you will have to wait for 3 or 4 weeks in the mouse model. So it is cheaper, faster than the mouse model, and the advantage is that you don't have to use the animals. So these models are helpful and are useful to analyze different biological processes like tumor growth, invasion, to make toxicity assay with new drugs, but also to control the activity of a new therapeutic agent. So in this case, if you want to focus on immunotherapy, these models have been validated to for detection of chicken CD8, CD3, and CD68 cells. And in fact, in the previous studies using B-cell lymphoma model, so this is a Xenoblast model using CD4 cell lines treated with anti-PD-1 antibody. So they have shown that the tumor growth is significantly inhibited in relation with the reintroduction of TILs within the cells detected by the labeling of CD3 and the CD4 membrane receptors. So in conclusion, we have been working on a new CD47 and CD19 B-specific antibody, so TG81. This antibody is showing very nice activity in different models of B-cell lymphoma, both in vitro and in vivo, and this activity is related to the increased ADCC and phagocytosis activity in the co-culture models in combination with the rituximab and various combination, the U2 regimen. So within this combination regimen, the new generation CD20 antibody rituximab is exerting stronger activity, both phagocytosis and ADCC, when compared with the standard anti-CD20 antibody, rituximab. we have shown that the relation of cytoskeleton may be associated with the synergistic activity of both approaches in the preclinical model of Burkitt lymphoma. And this remodulation of cytoskeleton may also help in favoring the infiltration of the tumor by NK cells and macrophages. And in link with this project, so this antibody is currently in the phase 1 clinical trials in refractory B-cell lymphoma. So as a conclusion, I would like to thank the members of my labs who have been involved in this project, mainly Marcelo, Diana, Mark, and Gianna, which were working hardly on this work. The collaboration with the group of Pablo Mendez was crucial for obtaining these BTK mutants. So with the work of Richard in the lab of Pablo. So this project was initiated in collaboration with the group of Francis Bosch at Vitro. And of course, we are acknowledging the support and the providing of the different drugs by TG Therapeutics and for Emmanuel Norman, which is coordinating this project together with me. So thank you very much, and I will be happy to take the question.
Hello, Gael. Thanks for the talk. It's been really interesting. So there are some questions here in the chat. So let's start with the first one. Has the CD47 blockade been tested in the aggressive double or triple hit lymphomas, which shown c-MYC overexpression?
So this is a very interesting. It's a great challenge because the double hit lymphoma are thought to be to not be dependent on this infiltration of human effectors. So due to this removal, which is characteristic of these cells, the oncogenic signal is thought to be enough to promote the growth of tumors. So they are not thought to be so dependent on macrophages, for example. But we have some evidences that it should not be the case. So we do believe that some tumor microenvironment-related signalings may be important for DHL progression. So it's something we're going to do. We're going to test this antibody in DHL preclinical models.
Another one, have you looked at what's happening with the other markers participating in the immune checkpoint in aggressive B-cell lymphoma? And how are other cells from the immune system being affected by these drugs?
Well, in fact, we mainly focused on B cells. It's true that it's something, well, something we can do because we are collecting the non-B cell fraction. So maybe for further genomic or proteomic analysis, we can do that. But so far, we are mainly focused on the CD20 positive fraction. So everything I've shown in the presentation has been made, any genetic approaches have been made following CD20 purification. But it's something we have to analyze, yes.
The other thing, have you tried to study the aberrant glycosylation of CD47 and how this mechanism can challenge aggressive B-cell lymphoma, immune evasion, and targeting?
Well, it's a very nice question. So I don't know. I imagine this could affect the stabilization of the receptor. So in the first study, when we were analyzing the direct effect of CD47 negation on chronic lymphocytic leukemia B cells, we used plenty of inhibitors targeting everything within the cells. And I think that no inhibitor, so the sole inhibitor which were able to modulate the transduction of the signal of the death signal induced by CD47 were those related to TPCK inhibitor and the depolarization of actin-like cytokeratin D. So everything related to post-transcriptional modification didn't induce any significant downregulation of the response to the antibody. So in fact, I'm not sure this would have a relevance for the transduction of the signals within the cells. So whether it can affect the relation with the macrophages, so it's still something that we have to investigate.
So another 1 trillion clinical trials was on hold last month due to grade 3 and 4 thrombocytopenia. I don't know if you're aware of that. How would you refocus this approach?
Yes, but I imagine you are talking about a pure CD47 antibody. So that's the reason why we're interested in this bispecific CD47 and CD19. So this kind of side effect is bound to be avoided by this double targeting.
But the fact that CD47 and CD19 are both expressed on healthy cells, would you expect any side effect?
Oh, yes, on the B cell compartments, yes, of course. But I mean that we are not expecting any effect regarding, or very low effect regarding platelets or red blood cells.
Okay, so I have another question. This is a little bit long. So in the mouse model, some mice treated with the triple combination were tumor-free, some with a smaller tumor and other with large tumor. Because those mice had the identical genetic background, is it possible that all the mice will eventually have big tumor if they are maintained longer? In other words, in other words, the treatment may delay tumor growth but not eliminate it. Can it be a possibility?
Yes. Yes, you're right. I think that we were unable to detect the tumors by classical histological examination, but of course, so I don't think that almost all the tumor B cells are deleted within this model, but I think that they are reaching undetectable levels. So this is the main advantage of the triple combination. We did not eliminate the tumor, but we are able to control the disease. This is the main message.
Okay, so if there is no more question, I would like to ask you again your talk. It was really interesting. Thank you, everybody in the audience. Thanks for the question, and I think we can finish here.
Thank you. Thank you so much.
Thanks.
Thank you. Goodbye.
Bye.